The identification of the bacteria involved in animal pathologies leads to the isolation of one or several pure cultures from organ lesions. Each bacterium is characterized by its morphology, its biochemical properties and ability to grow on specific media. The main interest of using anti-sera for coagglutination tests is to serotype the strains. Indeed, knowing the serotype of a strain is fundamental for the epidemiology of a disease and for the implementation of an autogenous vaccine, as there is generally no immune cross protection between serotypes. The anti-sera are proposed in 1 or 2 milliliter vials. The required equipment is very easy. A very clean glass sheet including discs, which is better than a small glass slide. The anti-sera or coagglutination reagents, physiological water, a pure culture of the bacterium to be serotyped, a Pasteur pipette, and a timer. Before testing, bacterial culture must be carried out overnight, or longer depending on the strain, at 37 degrees on a non-selective medium, generally on trypticase soy agar. Individual cases are described on the technical data sheet. All components should be brought to room temperature 20 degrees to 25 degrees before use, and excessive heat from the light source should be avoided. The vial must be vigorously shaken before use in order to thoroughly reconstitute the suspension. Both reagent and bacterial culture must be controlled before use. For the control of the reagents, dispense a drop of reagent, about 30 microliters on a disc. Then add a drop, about 30 microliters, of physiological water to it. The reagent is valid if no agglutination must be observed after mixing. If an agglutination is observed, this means that the vial has not been thoroughly shaken. Control of the bacterial culture. On another disc, dispense a drop, about 30 microliters, of physiological water. Pick off 3 or 4 colonies to be tested with a Pasteur pipette. The bacterial mass must be visible on the pipette. Carefully emulsify it in the water. The bacterial culture is valid if no agglutination is observed after emulsion. If there is an agglutination, the strain is auto-agglutinable and the test cannot be implemented. The test may be carried out once these 2 negative controls are validated. Dispense a drop of reagent on a new disc of the glass sheet using a Pasteur pipette. Pick off a few colonies. The bacterial mass must be visible on the pipette. Mix them with the reagent during 5 seconds. Then, gently rock and rotate the sheet to ease the agglutination process. The mixing process must last for 30 seconds. Reading should be done right after mixing. A positive result is characterized by the presence of colored agglutinates after 30 seconds of mixing the bacterial culture and the reagent. A negative result is characterized by no agglutination after 30 seconds of mixing the bacterial culture and the reagent. After the identification of an avian E. coli strain, the research of the serotype will be implemented by the Rapid Plate Agglutination technique with the Biovac avian anti-sera 01, 02 and 078. A drop of each reagent is dispensed on the plate. A few colonies are picked off and mixed to each reagent. plate is gently rotated for 30 seconds. In this case, there is no agglutination between the strain identified as E. coli and the antiserum E. coli 01 and 02, and there is an agglutination between the same strain and the antiserum E. coli 078, so the E. coli strain is serotype 078. In case of cross-reactivity, tests should be carried out again with a bacterial suspension in a saline solution. With an optic density between 1.4 and 1.8 at a 580 nanometer wavelength. This is equivalent to Macfarlane scale number 3. Mixing reagent and suspension lasts for 2 minutes. If there is still a cross-reactivity, dilute the suspension at 1/2 and 1/4. The serotype of the strain is the one where agglutinates appear first. In case of any doubt, Biovac can control the strains for you. A large range is available. Please contact us if you have any queries.